Calcium release is the basis of excitation contraction coupling. But how does it take place? Is it different for different muscle groups? Muscle cell, does it have any kind of structural support meant for this excitation contraction coupling? What is calcium-induced calcium release? All of these queries have been resolved in this video. Let's watch it and get benefit of it. Excitation and contraction coupling means what? Excitation is the generation of action potential. When we say any membrane becomes excited, that means the action potential is generated on that cell membrane. So action potential, a kind of spike potential is generated if we talk about the skeletal muscle. So this spike potential generation is the excitation of the membrane, and when membrane becomes excited, it will lead to the next response that is called mechanical response, that is your contraction. And how these two things, excitation means generation of action potential leading to the contraction. What is the mediating agent? That mediating agent is single, one and only, that is calcium ion. If the calcium ion in the cytoplasm of that muscle is in higher concentration, it will lead to the contraction. And through the mechanism of excitation, the higher availability of calcium ion leads to contraction, and this whole mechanism is called excitation contraction coupling. So basically, in a muscle cell cytoplasm, there is a calcium ion concentration available in this much of concentration, that is 10 raised to power minus 7 molar, and it will become higher 500 times and reach to 2 into 10 raised to power minus 4 molar concentration for the contraction to be generated. So at this concentration of 10 raised to power minus 7 molar, the muscle is in relaxed condition. But when the concentration reaches 500 times higher and calcium ion will become 2 into 10 raised to power minus 4 molar, the contraction will take place. Now, excitation contraction coupling is this mechanism that how this action potential leads to the increased availability of calcium ion concentration. This whole mechanism is discussed as excitation contraction coupling, because once the calcium ion is available in higher concentration, it will lead to the contraction. So calcium ion kahan se badhega, kaise badhega? Through the generation of action potential is discussed as excitation contraction coupling. Before moving to the excitation contraction coupling mechanism, let's first discuss the structural aspects of it. What are the structural support mechanisms present in a muscle for the development of excitation contraction coupling? So in this diagram, you can see here, this is the structure of the skeletal muscle shown here. Skeletal muscle, we have the typical band formation, A band and I band, that is the arrangement of your myofibrils, actin and myosin, and this band formation is present in the skeletal muscle. Along with that, here in this diagram, some tubular network is shown. That is called the sarcotubular system. Sarcotubular system has two types of tubules present in the muscle structure, that is the T-tubule and L-tubule. T-tubule is what? This is the sarcolemma running the cell membrane of a muscle cell, and this sarcolemma invaginates and it inserts into the cell itself transversely and then coming back and then running again on its surface and then invaginates inside transversely and then coming back. So these inversions of the cell membrane, muscle cell membrane, is known as the T-tubule. T stands for transverse. And along with that, here are some parallel tubular networks present, which are the part of the sarcoplasmic reticulum. So these tubules are called as L tubules. The L stands for longitudinal. So longitudinal tubules are the parts of the sarcoplasmic reticulum. T tubule is the invagination of the cell membrane itself. This T tubule, normally it is present here near the Z disc structurally, and along with in close proximity with L tubules. These L tubules as they reach towards the T tubules, यहाँ पे इनका they becomes the merging happens with each other, and here are the sacs formed, which are called the cisternae, terminal cisternae. So terminal cisternae, L tubule, then terminal cisternae, then T tubule, then next terminal cisternae and L tubule. So this is forming a unit required for the mechanism of excitation contraction coupling. So here this is called the sarcotubular system. The sarcotubular system, if we compare between among these three muscle groups, like skeletal muscle has very extensively well developed sarcotubular system, whereas in cardiac muscle it is less developed as compared to the skeletal muscle, and in the smooth muscle it is very less developed. It is least developed in the smooth muscle. So sarcotubular system is your structure present for the generation of excitation contraction coupling. Now how it works. So now here we are discussing the excitation contraction coupling mechanism in skeletal muscle first. तो skeletal muscle में क्या है कि excitation हुआ means action potential generate हुआ, ये आपका neuromuscular junction है, यहाँ पे acetylcholine release होता है, acetylcholine is going to come here binding to its receptors, then ligand gated sodium channels will open, after that voltage gated sodium channels will open, the sodium will enter in and it is going to produce the depolarization, or the action potential, the spike potential is generated. तो ये यहाँ पे एक्शन पोटेंशियल जनरेट हुआ है, ये सरफेस पे जनरेट होता है, ये जो आपका इन्वर्जन है, इन्वेजिनेशन है, दैट इज योर टी ट्यूब्यूल. तो ये एक्शन पोटेंशियल जाता है टी ट्यूब्यूल की मेम्ब्रेन पे, वेयर वी कैन रिप्रेजेंट इट एज इनसाइड पॉजिटिव एंड आउटसाइड नेगेटिव. सो यहाँ पे टी ट्यूब्यूल के मेम्ब्रेन पे एक रिसेप्टर प्रोटीन है जिसको बोलते हैं DHP, डाईहाइड्रोपाइरिडीन रिसेप्टर. दिस इज अ वोल्टेज सेंसिटिव रिसेप्टर प्रोटीन. It is linked with one more receptor ligand gated actually channel, which is called rhinodine receptor, and it is basically a calcium channel. So here the DHP receptor and rhinodine receptor, these two are acting as a unit which are physically interlinked with each other. तो ये वोल्टेज सेंसिटिव रिसेप्टर है, तो जैसे ही यहाँ पे डीपोलराइजेशन आता है मेम्ब्रेन पे, टी ट्यूब्यूल की मेम्ब्रेन पे, वैसे ही देयर इज सम कॉन्फिगरेशनल चेंज हैपेंस इन दिस DHP receptors which will lead to the opening of rhinodine receptor, which is a kind of calcium channel. So as this rhinodine receptor opens, this rhinodine receptor is present in your terminal cisternae, which is the terminal portion of your L tubule. And L tubule is a part of your sarcoplasmic reticulum. Sarcoplasmic reticulum is the storehouse of calcium ion. So as there is an opening of this rhinodine receptor or the calcium release channel, the calcium will come out, and here is the increased availability of calcium in the cytoplasm now, which will lead to the contraction to happen. Once this calcium ion is present in higher amount, where it goes? It will go to the troponin. It will bind there at troponin C. Configurational change will happen. Then there is the orientation of this tropomyosin will be changed, and it will open the active site of actin filament, and then the actin myosin cross bridge will be formed, which will lead to the contraction of the skeletal muscle. So ये आपका हो गया calcium release, and this calcium release will bind to troponin, which will lead to the contraction. यहीं तक ही हमारा excitation contraction mechanism आपको discuss करना होता है. अब during relaxation what will happen? The diagram below you can see, as there is the repolarization, means there is no more impulses there. The repolarization, the membrane is getting back to its resting condition, outside positive, inside negative, it represented like that. And as this potential coming back to the resting level, there is no excitation of the DHP receptors. DHP receptors come back to their resting stage, and then it will close this rhinodine receptors too. So this rhinodine receptor of the calcium release channel is closed, and on the other side of the sarcoplasmic reticulum there is calcium pump is working, which is called SERCA, sarcoplasmic reticulum calcium ATPase pump, which will take back whatever the extra calcium is present here. It will be taken back with the utilization of ATP into the sarcoplasmic reticulum. Here in the sarcoplasmic reticulum, it has one more protein which is called calsequestrine. This calsequestrine is having very high affinity for calcium ion, and it keeps the calcium ion bound with it once it is present in the sarcoplasmic reticulum. So as there is the resting stage comes, no impulse is there, the DHP receptor will come back to its resting stage, which will close the ryanodine receptor. So calcium incoming is now restricted, and whatever the extra calcium present, it will be taken back with the utilization of ATP by SERCA pump into the sarcoplasmic reticulum. There the calcium ion binds with the calsequestrine protein. So this is the mechanism of excitation contraction coupling in the skeletal muscle. तो जब आपके एग्जाम में ये आएगा skeletal muscle excitation contraction coupling, this whole thing, what happens during the action potential and what happens after the resting membrane potential during relaxation, दोनों चीजें आपको डिस्कस करनी. So now in this segment we are discussing the excitation contraction coupling of smooth muscles. तो यहाँ पे सबसे पहली बात तो इसका जो स्ट्रक्चर है, स्ट्रक्चर ऑफ सार्कोट्यूबुलर सिस्टम, इट इज वेरी लेस डेवलप्ड. सार्कोट्यूबुलर सिस्टम इज वेरी लेस डेवलप्ड मींस हियर यू कैन सी कि ये जो सेल मेम्ब्रेन है स्मूथ मसल सेल की, सेल मेम्ब्रेन सिर्फ छोटे छोटे स्मॉल डिप्रेशंस हैं विच आर गेटिंग इनसाइड, विच आर कॉल्ड दी कैवियोलाई. कैवियोलाई आर दी रिप्रेजेंटेशन ऑफ द टी ट्यूब्यूल्स प्रेजेंट इन योर स्केलेटल मसल, जो पूरी विड्थ तक जाती है, लेकिन यहाँ पे ये छोटा सा डिप्रेशन ही है. तो and this depression caveoli, around that caveoli the tubular system of the sarcoplasmic reticulum present. तो ये इतना यूनिट रहता है sarcoplasmic reticulum sarcotubular system का in smooth muscle. So smooth muscle का जो calcium ion availability है during its contraction, it is not only dependent upon the calcium ion present in the sarcoplasmic reticulum storehouses. It has to take the calcium ion from the extracellular fluid, and for that here in the central diagram you can see that as the action potential reaches here, okay, as there is the change in the membrane potential, it becomes depolarized, outside negative, inside positive. As this depolarization present here, it will, what it does, it will open the voltage sensitive calcium ion channels which are present in the cell membrane itself. So these are the voltage sensitive calcium ion channels. Calcium is more of course in ECF as compared to ICF. तो ECF से ICF में calcium आना शुरू हो जाता है. दूसरी तरफ ये जो potential change है, ये sarcoplasmic reticulum में भी calcium channel को open करता है, as we have seen in case of skeletal muscle. जो कैवियोलाई के नीचे जो सार्कोप्लाज्मिक रेटिकुलम का पार्ट है, so where this is the coupling of your dihydropyridine and ryanodine receptors, which are very less developed, of course, less in number, but that is present. That mechanism is also present in smooth muscle. So this potential change will lead to the calcium ion calcium channel opening in sarcoplasmic reticulum, and calcium will be coming out from the sarcoplasmic reticulum. This calcium is coming from ECF, this is coming from this sarcoplasmic reticulum, and this calcium is now in increased concentration. This calcium will bind to calmodulin protein, whereas in skeletal muscle we have seen it was troponin. Here in smooth muscle it is calmodulin, and calcium calmodulin complex ultimately going to produce the phosphorylation of myosin, which will lead to the muscle contraction. So जब हम एक्साइटेशन कॉन्ट्रैक्शन कपलिंग पढ़ते हैं तो हम यहाँ तक पढ़ते हैं कि कैल्शियम आयन बढ़ गया. अब ये कैल्शियम आयन जाके कौन से रेगुलेटरी प्रोटीन के साथ बाइंड करेगा? इट विल बाइंड टू द कैल्शियम इट विल बाइंड टू calmodulin protein, and this calmodulin complex, calcium calmodulin complex, will ultimately activate the contraction mechanism. Now in the right side of the diagram, the relaxation is shown. During relaxation, what will happen? Here the membrane potential will coming back to its resting stage, and Here what will happen? The calcium ion will have to go back into ECF and into sarcoplasmic reticulum, and with the help of pump, ATPase pump, both the sides, the calcium ion will be taken back. So this increased calcium will go back into the sarcoplasmic reticulum with the help of ATPase pump, and it will go back into ECF with the help of this calcium ATPase pump. And here, because of the less availability of the calcium ion, this complex is dissociated, and that will lead to the dephosphorylation of the myosin chain, which will lead to the relaxation of the muscle. Here, one point has to be discussed, that the voltage gated calcium ion channels which are present here in the cell membrane, which will make the calcium ion availability inside the cytoplasm, these are slow opening channels. Okay, so slowly opening channels, this will remain open for a longer duration of time. So in smooth muscle as compared to skeletal muscle, the calcium availability is prolonged and the contraction will also be prolonged. So now we are moving to the next segment where we are discussing the excitation contraction coupling in cardiac muscle. So regarding the structural system, that is our sarcotubular system. In cardiac muscle, the sarcotubular system is not as developed as skeletal muscle, but it is more developed than the smooth muscle. And here what happens when the action potential reaches on the cell membrane surface of myocardium. This is the action potential which is in the form of plateau potential here, and this action potential reaches on the surface and then it going down towards the T-tubule. This T-tubule is not as much deep as in the skeletal muscle, but it is present there. This T-tubule, as the potential reaches here, this is outside become negative and inside becomes positive. Here the L type of calcium channels will open, which are the voltage sensitive calcium channels. L stands for long lasting here. And here this calcium channel will open. This calcium will enter into the ICF in the myocardial cell from ECF. So from ECF, here the calcium is present in abundant amount as compared to the ICF. The calcium is moving, diffusing inside the cell of myocardium. And in the sarcoplasmic reticulum part, that is a L tubule of this sarcotubular system, uska jo cisternae hai, this is having one more type of receptor channel protein, which is again rhinodine receptor, as we have seen in case of skeletal muscle. This rhinodine receptor is present in the terminal cisternaes of the sarcoplasmic reticulum, and this rhinodine receptor is sensitive for the calcium ion coming from ECF to ICF. And this calcium ion, what it is going to do, this calcium ion is going to open this rhinodine receptor channel, and the calcium will enter from sarcoplasmic reticulum towards the cytoplasm. Okay, so this is called calcium induced calcium release. Which is typically seen in case of cardiac muscle. So the calcium, as there is action potential, reaches towards the T-tubule. Here the calcium is present in ECF. So this calcium will enter, this calcium channel, long lasting calcium channels will open because of the voltage change. The calcium will enter from ECF to ICF. This calcium is available for muscle contraction. Also this calcium will lead to the opening of rhinodine receptor calcium channels, and this calcium will enter from sarcoplasmic reticulum towards the cytoplasm. So ECF से भी आ रहा है calcium और calcium आ रहा है through sarcoplasmic reticulum by calcium induced calcium release, and this is called calcium spark, which is coming from sarcoplasmic reticulum, and this whole calcium which is available here, it can be utilized for contraction. And here the regulatory protein for calcium ion is again troponin, where it is going to bind. Once the calcium ion is present here in ICF in the cytoplasm, it will bind to the regulatory protein, that is troponin, and the same way as in the skeletal muscle the contraction begins, the same way it begins in cardiac muscle. Here on the right side of the diagram, the relaxation procedure is shown where the calcium has to go back to ECF and sarcoplasmic reticulum. So once there is no action potential present here on the cell membrane becoming repolarized and then resting membrane potential is achieved, then the calcium will be taken back into the sarcoplasmic reticulum by sarcoplasmic calcium pump, ATPase pump, which is called SARCA2 kind of pump is present here. So with the utility of ATP, the calcium will be taken back into the terminal cisternes of sarcoplasmic reticulum, and also this calcium which is coming, which is excess in here, it has to go back to ECF also, because ECF से भी calcium आया था. ये calcium ECF को जाता है with the help of calcium sodium antipode. Calcium will go out and sodium will come in. Sodium is coming in because here the sodium ion concentration is accumulated because of the sideline working of this sodium potassium ATPase pump. So sodium will be sent out, it will be accumulated here in the ECF at this point, and the sodium will bind to the transporter protein here, and calcium will bind here to the transporter protein. The calcium will be thrown outside, sodium will be taken inside. It will be recycled through this sodium potassium pump back, sodium will go back, it will come here inside, and calcium will be moved outside. So this is a kind of, which kind of transport? This is called secondary, an example of secondary active transport, where the energy is utilized by sodium potassium pump and calcium is transported outside the cell, myocardial cell. So this is the whole process of excitation contraction coupling discussed in case of cardiac muscle. Last segment of our video. Here we are going to compare the excitation contraction coupling features in all three different muscle groups, in skeletal, smooth and cardiac. So this is a kind of summary section of our video. So here, features of excitation contraction coupling written, the calcium requirement. All three muscles for excitation to contraction, action potential leading to contraction, it needs calcium ion. So all three muscle groups, only one agent is required, that is calcium ion. Sarcotubular system, that is the structural system required for excitation contraction coupling, where it is going to release the calcium ion into the cytoplasm. So sarcotubular system is very well developed in case of skeletal muscle, very less developed in case of smooth muscle, and here it is not so well, but it is there in case of cardiac muscle. Calcium source. Source of calcium coming inside the cytoplasm, it is solely through sarcoplasmic reticulum. There is smooth endoplasmic reticulum which is called sarcoplasmic reticulum of skeletal muscle, terminal cisternae of it, which are the storehouse of calcium ion. The calcium is released out from the terminal cisternae of sarcoplasmic reticulum. In smooth muscle, the source of the calcium ion mainly it is ECF, and some part of it is coming through sarcoplasmic reticulum. In cardiac muscle, the calcium is coming from ECF and sarcoplasmic reticulum. So here in case of smooth muscle and cardiac muscle, the calcium ion concentration in ECF is going to affect the muscle contraction. Whereas in case of skeletal muscle, the concentration outside, the concentration of calcium in blood or in ECF is not going to directly affect the mechanism of contraction, because that is solely dependent on the availability of calcium in the sarcoplasmic reticulum. Calcium induced calcium release. This is the typically feature of cardiac muscle. It is not that much present in case of smooth muscle, and it is not present in case of skeletal muscle. Regulatory protein. Regulatory protein means once the calcium is released out into the cytoplasm, where it is going to bind with. So in case of skeletal muscle, it is the regulatory protein is troponin. In case of smooth muscle, the regulatory protein is calmodulin. And in case of cardiac muscle, it is same troponin. Relaxation. During relaxation, how the calcium is taken back. Relaxation में, in case of skeletal muscle, there is the SERCA pump, sarcoplasmic reticulum calcium ATPase pump, where the calcium is taken back into the terminal cisternes of sarcoplasmic reticulum. In smooth muscle, the calcium is moved into the sarcoplasmic reticulum through the ATPase pump, and same way it will move into the ECF with the help of calcium ATPase pump. In cardiac muscle, the calcium is moving into the sarcoplasmic reticulum with the help of SERCA2 pump. Also, the calcium will move out of the cell into ECF with the help of calcium sodium antipode, which is a kind of secondary active transport that we have already seen. The duration of the calcium pulse. What the calcium pulse means? Calcium pulse is the rise in the calcium ion concentration as the action potential reaches, and during relaxation there is again coming back to the relaxed state. So duration of this whole cycle of the calcium ion, which is very less in case of skeletal muscle, it is around 1/20th of a second. For that duration, the calcium will be remaining in the cytoplasm, otherwise it will be taken back. In case of smooth muscle, it is very prolonged. It goes up to few seconds. Means calcium will remain in the cytoplasm of the smooth muscle cell for few seconds even. And calcium pulse duration in the cardiac muscle, it is longer than the skeletal muscle. Okay, because there are also the calcium ion present because of the action potential, which is a kind of plateau potential. So its potential itself is very long. So it will promote the calcium remains there in the cytoplasm, myocardial cell. So this is all about the aspects, all details of excitation contraction coupling in different types of muscle groups. Hope you find this video helpful. If yes, then do not forget to subscribe my channel and share this video to your friends, those who may need it. Bye-bye.